Hello, everyone, and welcome to today's live broadcast, the Personalis ACE CancerPlus test, a comprehensive DNA and RNA sequencing solution for guiding clinical care and improving patient outcomes, presented by Heather Wetzel, MS, LC-GC. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by Personalis. Personalis Incorporated provides researchers and clinicians with accurate DNA sequencing and interpretation of human exomes, genomes, and transcriptomes. The company supports researchers and clinicians engaging in clinical trials, case control, or family-based genomic studies of disease and cancer. Its ACE, accuracy and content enhanced technology, supplements a standard exome, substantially increasing its medically relevant coverage and accuracy. Personalis builds on this enhanced sequencing foundation with innovative strategies for alignment, variant calling, annotation, and analysis. Through this comprehensive approach, the company provides genomic data and interpretation of the highest accuracy. Its in-house laboratory is CLIA licensed by the State Board of California, Florida, Maryland, and Pennsylvania, and is CAP accredited. To learn more about Personalis, please visit www.personalis.com. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want, anytime you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on your screen. We'll answer as many questions as we have time for at the end of the presentation. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CME CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. Finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or you can use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Heather Wetzel. Heather is a field applications scientist and genetic counselor at Personalis. She received her master's in genetic counseling from Northwestern University in 2006 and board certification in genetic counseling in 2007. Prior to working at Personalis, Heather worked for 8 years as a clinical genetic counselor in specialties including pediatrics, cancer, and prenatal at the University of Illinois, Northwestern, Stanford, and the University of California, San Francisco. Heather currently supports the clinical products at Personalis, including the ACE clinical exome for Mendelian diagnostics and the newly launched ACE CancerPlus test for tumor profiling. Please join me in welcoming Heather Wetzel. I will now turn the presentation over to her.
Thanks so much, Judy, and hi everyone, good morning. Thanks so much for joining us. We're excited to talk about our newly launched ACE CancerPlus test. And so without further ado, I'll go ahead and get started in with the presentation. And so first, I want to go over the learning objectives just to sort of review what we're going to talk about today. So first, the purposes of the presentation are to be able to understand some of the current industry challenges with regard to next generation sequencing and tumor profiling for clinical care. Second, to appreciate how the use of Personalis' accuracy and content sequencing platform, or ACE, with DNA and RNA analysis improves precision in reporting in solid tumor testing. And third, to recognize through case studies some of the more common molecular changes that are identified that might assist in therapeutic management. What I hope to cover in today's session is a few of the topics listed here, starting with a review of the sort of high-level overview of the CancerPlus details, as well as a short background about Personalis as a company. And then we'll transition to talk about some of the current industry challenges that face clinical next-gen sequencing cancer testing. And alongside that discussion, we'll talk about some comprehensive solutions that are offered with the ACE CancerPlus test and sort of what we've done to address some of those challenges. We'll follow that up with a couple of case studies. And then in the session, talking about some logistics of the ACE CancerPlus test, including some documentation that we have available for patients as well as physicians, and then information on ordering the CancerPlus test itself. And then, of course, time allowing, we'll have some Q&A at the end. And so to first start, to talk a little bit about Personalis and our company, Personalis was founded in 2011. We're a company based out of Menlo Park in California, about an hour south of San Francisco or so. And the services that we offer are all built on this ACE platform. And so you'll hear us, when we talk about our different products, use the term ACE platform quite a bit, because everything that we do at Personalis is really built upon this accuracy and content enhanced platform. We have two different arms of services or tests that we offer through Personalis. On one side, we have a number of research services. So as you see here, some large-scale sequencing analyses for complex disease, Mendelian genetics and inherited disease research. And one of our bigger focuses is cancer research. And so we engage with a number of customers for clinical trials, translational research, as well as immuno-oncology, which is one of the hotter topics late last year and, of course, this year. What we'll talk about most today is really the clinical side of the products. And, those clinical products, of course, are what you see listed here, our ACE Clinical Exome Test, which is our test for Mendelian diagnostics and so-called diagnostic odysseys, and another clinical exome test that we've done through a partnership with Athena Diagnostics called the Neurome. Of course, for today's purpose or today's presentation, we're really going to talk about tumor profiling. And with tumor profiling, that's our ACE Clinical Exome Test, our ACE CancerPlus test. And so just to go over a few sort of higher level details of the CancerPlus test, the test itself, again, powered by the ACE platform that we've talked about. The test consists of reporting of 181 genes of clinical relevance. That test is sort of powered by a larger extended cancer panel of over 1,300 cancer genes that we're interrogating. And that data, the raw data of that 1,300 cancer genes, is actually available upon patient consent. However, the actual interpretation and reporting is based on a much smaller subset of those genes, that 181 list. The test includes DNA and RNA analysis of solid tumors. And the test, by doing DNA and RNA analysis, we're able to identify a number of different types of variants, including base substitutions, insertions and deletions, as well as copy number alterations and gene fusions. Those 181 genes that we're reporting are genes that we're reporting based on their relevance to having associated FDA-approved therapies or clinical trials that might be available for a number of those 181 genes. And of course, by looking at a smaller subset of genes with this technology, we're able to obtain a very high sequencing median depth of approximately 500x, and that really gives us the ability to detect some of these low frequency variants, which we know are important, particularly in the cancer test or in the cancer setting. And so some of our early testing using the ACE CancerPlus test has really been through our relationship with the Department of Veterans Affairs. And so Personalis, just as a little bit of background, we've had a very long-standing relationship with the VA. The VA was one of our very sort of early customers in terms of partnering with them to do whole exome and whole genome sequencing as part of their Million Veterans program. And we've also sort of, along with that study or following those studies, have engaged with them with another of other types of tests. One being, our clinical exome test for diagnostic odysseys, and then the other being, working with the VA in Boston to pilot our ACE CancerPlus test. And so, that's a test that we've been working with them on for a few months now, and sort of in the process of a national rollout with the VA. And what you see on the slide here is really a quote from one of the directors of that program, Dr. Lou Fiore. And Dr. Fiore has been really, of course, impressed with the findings from the CancerPlus test, and has sort of said that the majority of the patients that they've tested, which primarily are going to be non-small cell lung cancer patients, but the majority of them who they've tested have actually had variants that have been associated oftentimes with an FDA-approved therapy. And so they're really using this sort of information to really help further develop their precision medicine program and to make some of these drugs more available to veterans nationally. So what I want to talk about next is really just sort of reviewing and talking about some of the current challenges that are available, that are sort of exist in the clinical world in terms of next generation sequencing cancer testing. And alongside talking about that, we'll talk about some of the solutions that are offered with the CancerPlus test. So if we think about some of the challenges that are out there, a number of these challenges are probably familiar to you if you're a clinician potentially ordering some of this testing. And so just to sort of walk through them, one of the main challenges that we talk about or hear about is really having a poorly focused gene test. And so some laboratories may have a large number of genes, hundreds of genes on their panel. Some may have less than 100 genes on their panel. But the idea is that oftentimes those panels either have too few or too many genes that aren't clinically focused, which is what you really need in this particular setting. Coverage is also an issue. Of course, with next generation sequencing, it's notorious for having some sort of areas of the genome that are really difficult to sequence, and that applies to exome sequencing, but of course it also applies to the cancer setting. Limited analysis of gene fusions is also an issue because not a lot of laboratories are doing RNA sequencing as part of their analysis, and so the ability to detect gene fusions is somewhat limited and oftentimes not available or not able to pick up a large number of fusions or newer novel fusion events. Of course, FFPE challenges, we'll go into that in more detail. And then the last couple, one, assessing platform accuracy and sort of how do we validate the testing that we're offering with the lack of gold standards out there in the field. And then how do we sort of translate all of this sort of information that we have at the laboratory level into a report that's helpful for a clinician to read and clear and concise. And so what we've done is really taken some time to address these sorts of issues. And of course, these issues are not all inclusive. There are a number of other issues out in this sort of space, but these are some of the ones that we've really, taken the time, we're taking a step back to really address those issues with our analysis. And that we now have what we feel is a more clinically focused gene set, better coverage. We've incorporated DNA and RNA analysis. We've improved our capabilities for FFPE sample analysis. We've validated our test. And we've written and made a report that's really concise and intuitive, that's really built for clinicians. And so what we'll do next is sort of walk through each one of these and give a little bit more information or more flavor for what we're talking about there. And so first, to talk about the gene set itself. So I mentioned briefly, that it's really, difficult for laboratories to sort of have a well-focused or clinically focused gene set because there's so many different cancer genes out there, and trying to decide what should go on a clinical cancer test has oftentimes been difficult. So the goal at Personalis was really to keep the CancerPlus test clinically focused. Of course, the test is based on a much larger panel, and that panel, that raw data is available for individuals looking for, say, translational research, for example, and wanting more information about a larger number of genes. But we've really sort of honed in for the clinical reporting of those 181 genes to make sure those are genes that are clinically relevant. And so those genes themselves, the vast majority of them are genes that are associated with active clinical trials or FDA-approved therapies. There is a subset of genes on that list in our efforts to future-proof what we're doing and what genes we're including on our CancerPlus tests. Genes that are frequently mutated in cancer, but they may not have a current therapy that's associated with them, but perhaps they will in the near future. And so some examples of that are what you see listed here. So the CDH1 gene that's oftentimes thought of as associated with gastric cancer, as well as JAK1 and RB1 are just sort of three examples of genes that are also included on that panel. So next I want to talk a little bit about gene coverage. And again, this is something that, we talk about quite often in, sort of the exome sequencing setting, but this also applies to panel testing and cancer testing as well. And so what I want to do is sort of walk you through what you're looking at here on this particular diagram. And so in yellow, these rectangles that you see here are really representing, DNA target regions or exons, for example. And what you see in blue might be what a standard panel test could pick up. And so you'll oftentimes see that some exons might not be well covered, some may be incomplete, and some may not be covered at all. That's sort of where our ACE supplementation on the sequencing side really comes into play here, because we have developed in-house, a very specific chemistries and probe designs that have allowed us to be able to capture a number of these regions that are poorly covered by standard technology. And of course, when you're looking at, sort of a more focused gene panel, you're able to obtain better sequencing coverage than you would, say, with whole exome. But oftentimes, some of these areas that are difficult to sequence, with exomes, such as areas where there's high GC content, are still going to be difficult to sequence with a panel. And so that ACE augmentation really supplements those areas where there's poor coverage. And so what you end up seeing is really a combination of what, sort of might standardly be available, plus what would be available with the augmentation that's provided by that ACE platform that builds our products. Those areas, that are targeted, are what you see listed here. And so I've mentioned high GC regions. That's one of the more common areas that, is supplemented by ACE technology. But we're also looking outside of the exons as well. Of course, there are medically important variants that aren't in the exons. And so we're also targeting known intronic variants or other biomedically important regions that are outside of the exons. And of course, filling in those gaps in those medically important regions. And so this sort of ACE augmentation is really what builds sort of the platform itself and improves the sequencing coverage for all of our products, including the ACE CancerPlus test. And so when you see, how that might look when you're thinking about individual genes, what I've put here are just a couple of gene examples of genes that are part of the clinical reporting for the CancerPlus test. And so these are probably genes that might be familiar to you. So AKT1, for example, is a gene in cancer that's associated with a large number of clinical trials. And then CDKN2A is one of the more frequently mutated genes in cancer. And so this is a very common gene for which we find variation on these clinical tests. And so what you're looking at here along the y-axis is the depth of coverage, or sort of how many reads that gene or that sort of coding base has. And what you're seeing along the x-axis is your coding regions of this particular AKT1 gene. And so what this is really showing is how comprehensive the coverage is for these particular genes. And again, these are just two examples. We certainly have examples of all of these genes that are on this panel, but this is really just speaking to that sort of comprehensive coverage that is addressed with that ACE platform. So we've also published on our technology. We released a publication in August of last year in Genome Medicine, and the publication itself was written by Personalis and co-authored with the NIST, or Genome in a Bottle Consortium group. And what we really were looking at was comparing our ACE platform to a number of sort of off-the-shelf or conventional exome sequencing kits, as well as looking at whole genome sequencing. And we were looking at sort of that comprehensiveness of coverage, or gene finishing, as we call it, and comparing our ACE platform to a number of other different tests that or platforms that are out there on the market. And so what we really found is that ACE has very superior coverage and an ability to finish a number of medically relevant genes. The figure that you're looking at here is one of the other areas that we focused on for the publication, and that was looking at that sort of issue of GC content. So I mentioned before that, high GC content is often a reason why next generation sequencing actually fails and is unable to sequence certain areas of a gene. But what this is really showing is that if you're looking at gene finishing or coverage on the y-axis, and the sort of degree or percent of GC content on the X, what this is really showing is that ACE in green has a really high level of gene finishing all the way across various sort of levels of GC content, so to speak. So even at a lower level, all the way up to, sort of 70–80% GC content, that platform, that ACE platform, is really able to sort of continue to sort of well sequence and cover these difficult regions, whereas some of these other platforms, as you can see, as the GC content starts to increase, the sequencing coverage really starts to fall off significantly, which is the case with some of these exome sequencing platforms, and of course the case with something like whole genome sequencing. So what I want to move to next to talk about is gene fusions and how sort of we've addressed the sort of issue or difficulty of accurately assessing gene fusions in clinical cancer testing. And so I briefly mentioned earlier when we were talking about sort of the high-level details of the CancerPlus test that we do include DNA and RNA analysis of those 181 genes. And so we're not, for this test, selecting a certain number of genes where there might be common gene fusions or known fusions. We're actually doing full RNA analysis of all of those 181 genes. And as a result of doing that, of course, we're able to detect gene fusions in all of those 181 genes on the panel, which would allow us to pick up sort of new fusion events. It also allows us to be able to pick up gene fusions in those 181 genes, but in those genes and other partner genes that might not be covered on that 181 gene panel. And so this is a great example of what I'm saying here. ALK, of course, is a common cancer gene, and it's associated with non-small cell lung cancer, but this is a very common fusion event involving the EML4 gene, which isn't a known cancer gene. Of course, it's not on any sort of common cancer panels, but it is a fusion that would be detected with our ACE CancerPlus test because we're including ALK and RNA sequencing to detect that fusion, and we would also pick up the fusion partner, this EML4. What you see here are really just a number of other examples of some of the gene fusion partners that have known targeted therapies available. So some of these, of course, ALK we've just talked about with non-small cell lung cancer, RET fusions and thyroid cancer, ROS1 fusion events and other mutations with lung cancer, and then the NTRK1 fusions as well. And so these are just some examples. Of course, there's a number of others, but this just sort of gives you sort of a flavor of why really complete RNA analysis is important, particularly in detecting gene fusions, which are oftentimes associated with with heme malignancies. But we're also, really quickly learning that, there's a large number of solid tumors that also have gene fusions as well. And so I also want to talk some about challenges with FFPE. And, again, if you're, a pathologist and you're ordering this sort of clinical testing, or even an oncologist, for example, who might be submitting this testing, oftentimes, what happens is you may submit a sample from FFPE tissue, because most of the patients who are undergoing this type of clinical testing, aren't necessarily patients with a newly diagnosed tumor where you have that fresh frozen tissue that you can submit. Oftentimes, these are individuals who are sort of much further along, unfortunately, in their cancer diagnosis, and this might be more of a, sort of third or even fourth tier test after some other treatments might not be working. And so oftentimes, clinicians are sort of going back into the archives and getting samples from FFPE that have been preserved. And oftentimes what happens is that those samples fail either at the QC step, even before sequencing, or we're just not able to get, really successful DNA sequencing, just because some of the issues with preparing and fixing with FFPE lead to degraded DNA. And so, of course, we really, rather than just sort of accepting that as a problem in the field, at Personalis, we really sort of took a step back and said, well, let's think of some ways to improve our capabilities with getting good quality data from FFPE. But since this is such an important concept, or, sort of tissue type in cancer. And so what this is showing here is some of the published data that's been looking at success rates with FFPE. And so, as I've already mentioned, those studies are sort of showing a sort of average success of around 30%. And this is one of the citations here from Hedegaard and all a couple of years ago. What we've seen through our own experiences with FFPE, just due to the sort of specific sort of technologies that we've developed in-house to improve our capabilities, is that providing, a good sample of a good tissue sample, we've been able to successfully extract and sequence approximately 90% of the samples that we've received that have been in FFPE tissue. And so, the sort of take-home message in this case is that, while FFPE tissue samples might be really difficult for some laboratories, we really haven't had, really any difficulties as compared to those laboratories. And while, of course, fresh frozen is always the preferred method or preferred sample type, we don't shy away from sequencing any samples that are provided through FFPE tissue. So what I'd like to talk about next is just a little bit of background about validation. And, of course, anyone who's looked at, other clinical cancer testing laboratories, of course, validation is a really sort of, I guess, difficult sort of concept, because there aren't any gold standards out there for us to really sort of assess our platform accuracy or compare them to. And so oftentimes what you're left doing is really, sort of establishing your own gold standards in-house and developing them that way and using that for a comparison. And so, what I want to talk about here is just some of the numbers behind our validation of the ACE CancerPlus test, keeping in mind, as I've mentioned again, while the CancerPlus test is really looking at those, approximately those 181 genes, the actual testing itself is based on that much larger panel of over 1,300 cancer genes, and that's what the validation is based upon. To actually validate that product or that test, we ended up procuring close to 40 different cell lines to be able to look at, different cell lines from different tumors, and also looking at various allele fractions, anywhere between 5% all the way up to 95%, to sort of have a very comprehensive or robust validation. And so, we were really happy with the results that we found from the validation study, and so some of those numbers are listed here. And what we found for base substitutions was over 99% sensitivity at approximately as low as a 5% allele fraction. For insertions and deletions, also over 99% sensitivity at an allele fraction as low as 10%. And also really great numbers for copy number alterations and gene fusions. And as you see here, high specificity as well, over 99% for base substitutions and indels as well. So last but certainly not least, in terms of talking about some of the challenges, I want to also talk some about reporting. And so again, once you've gotten to the point of, really improving the sequencing, improving your ability to detect fusions and get good, DNA from FFPE tissue and having that validation, all of that's really important. But at the end of the day, these are sort of analyses that are being reported to a physician who is then going into the room to sort of translate these results to their patient to determine if there might be any clinical trials or therapies that would be available in terms of their treatment program. And so it's really important that a report that any laboratory provide really makes sense and is as concise as possible. And oftentimes what we see is that these reports from this sort of clinical cancer testing are oftentimes 20 to 30 pages long because there's so much information to list and to detail. So it's really hard to sort of hone in on the important parts of a report. And so, at Personalis, we've really taken the time to do that, both with our clinical exome test that we do for Mendelian disease, and we've sort of translated that here to our ACE CancerPlus test with having a much more concise report. And so one of the parts that I want to sort of hone in on here, this is just a snapshot of the first page of the report, and obviously I don't expect you to be able to read every single word on it, but I just want to point out, on the first page, of course, you've got your summary of your patient information. And what I've got sort of circled here is a summary of the findings themselves. And so sort of right there, kind of in your face on the first page, is a nice summary, in this case, of the relevant variants that were detected in this particular patient who had a diagnosis of melanoma. And so, this list in this case, 3 different variants that were detected and 3 different genes on that first page. And then what you see on the following, at the end of this page and the following page, is really a short summary about those particular variants that were detected, just to sort of briefly go over what was found before going into a ton of other detail about the actual variant itself and its association with disease, as well as all the clinical trials. And so just to sort of point out what you're looking at here, this is that short summary that I mentioned of these 3 different variants that were detected. And so what we've provided on the report is a table that lists out the sort of availability of therapies that are associated with this particular variation. And so, just to sort of walk you through a couple of these, this is the very common BRAF V600K mutation that was detected in this case, again, a diagnosis of melanoma. And this is telling you here that there's an FDA-approved therapy that is associated with that particular tumor type. If this were no, then the next part is sort of, is there an FDA-approved therapy that's maybe not available for melanoma, but available for a different indication, and that may be able to be prescribed off-label, for example. And then here, are there any active clinical trials that are available for this particular gene or particular variant? And so in this case, again, with BRAF being a very common variant that's detected in cancer, and particularly melanoma, the answer to all of these questions is yes. So you would imagine that on the pages following on the report, there's probably a pretty long list of some of these FDA-approved therapies and clinical trials. A little bit of a different scenario with something like a P10 mutation in melanoma, where there's not a therapy approved for that particular cancer type, but there are FDA-approved therapies for other cancer types and active clinical trials. And so, following this summary, there's a lot more information on the following pages, but again, this is really meant to sort of be sort of a quick glance on that first couple of pages of what might be available for these different mutations, particularly when we're reporting more than one variant. It's important to have that summary information up front. And so now that we've talked about, some of these sort of industry challenges and what we've done at Personalis to really address some of these challenges, what I want to talk about next is just a couple of case studies that we've been able to identify from some of our earlier testing, and sort of how important this clinical cancer testing can really be. So in this particular case, this is just a very short, very straightforward case study. But what you see here is that in terms of the clinical presentation, this was a 65-year-old male who had been diagnosed with a lung adenocarcinoma. And the sample that was submitted to us for analysis was, of course, the popular FFPE with 50% tumor content. And we were able to successfully sequence that particular sample and resulted an exon 19 in-frame insertion or indel that was found in the EGFR gene. And this particular gene is another pretty common cancer gene. And this gene has a number of targeted available therapies, from FDA-approved therapies, as well as a number of active clinical trials that are associated with that particular variant itself. The second case study is interesting really from a sort of sample preparation and quality standpoint. This was another sort of typical presentation, in that this was a 56-year-old male who'd been diagnosed with an esophageal adenocarcinoma. And we ended up having to get a couple of different specimens from this particular patient. And so the first sample that was submitted to us was, of course, from the primary tumor was an FFPE sample. And we had extracted and sequenced a number of different times because we were having trouble at the QC step. And so we do a sort of QC sort of component even prior to sequencing, just to sort of do a check of the quality of the sample itself. And it continued to fail at the QC step for DNA. And so we, ultimately sort of checked back with the laboratory who had prepared the sample, and the specimen itself had been reportedly processed by microwaving by the submitting pathology lab. And so for, just for a non-pathologist, and this was something I had to kind of look into myself, because at first I was thinking, microwaving, that sounds crazy. Why would you do that? It's not unheard of in, other sort of preparations for processing samples. It's just not something that is, great to do in terms of sequencing, because it's going to really, affect the DNA quality itself. But it's sort of a great method of processing samples for other things, like maybe immunohistochemical staining or something like that. So anyway, so we were unable to sequence with that particular sample. And we had another sample that was submitted later on that was a sample from the metastasis of that cancer, submitted as fresh frozen tissue. And so with that sample, we were able to successfully extract and sequence the DNA and RNA. And so what you see here are some of the results that we had identified in this particular patient. Again, that common CDKN2A gene that has associated clinical trials, and an IDH1 splice site variant, as well as a TP53 mutation. So again, still some important findings for this particular patient, because this identified some clinical trials that might be available for him. Just as sort of a follow-up, sort of FYI, we did take a look further at that sample that had sort of failed the QC step. And even though we didn't submit results, and put this on a clinical report, we did, sort of complete the sequencing for this particular case, and those 3 variants that we found in the primary, in the sort of metastatic sample, we did identify them in that sort of sample from the FFPE tissue. So even though the sample had failed the sort of QC metric step, we still were able to have, the same results that we had found with the sort of secondary tissue. So the last part of the presentation is really sort of just going through some logistics of the CancerPlus test. And so we've prepared a number of different documents in an effort to provide as much sort of information to a clinician that might be needed to really sort of talk about the CancerPlus test and talk about tumor profiling with the patient. And so, of course, we have an ACE CancerPlus test sheet, and basically this really provides some of those higher level details that I've mentioned earlier on. It does include the list of those 181 genes that are part of the clinical test. We also have sort of clinically focused specimen preparation guidelines, both for FFPE and fresh frozen. So particularly if you're preparing samples, it sort of gives you guidelines for how to prepare those samples, but then also how to send those samples to our laboratory to sort of maximize all of our chances of success about getting reports from these samples. And then, of course, requisition forms are available and will eventually be available online, but they are part of our sort of sample submission kit that we provide to the clinicians. And so I have a picture of that specimen transport kit here on the next slide. And so, again, these are specimen transport kits that are sent to the clinician who is ordering the ACE CancerPlus test. And in that kit, is all the information that you would need, including the requisition form and ordering form, to be able to submit a sample to the Personalis clinical lab. A little bit more detail in terms of logistics. Of course, this is a clinical test. It's something that has to be ordered by a physician. At this point, we're set up for billing as a self-pay through a patient or institutional billing. And right now, the CancerPlus test is only available in North America. We do have plans to sort of extend our offering globally, but right now with sort of an early launch of the test, right now just in North America only. And so in terms of what's needed, everything's in that kit. Requisition form, transport kit would be mailed to Personalis, and the turnaround time for the test is approximately 3 weeks. Last but certainly not least, we've also developed a patient FAQ document. And the FAQ document is really just sort of thinking about some of the more common questions that might come up in a patient setting. And so this is meant to be something that can be provided to patients to sort of take home and read more about the ACE CancerPlus test, or it can be used for a clinician as really a guideline of what you might talk about with your patient in terms of ordering this type of testing, how long it takes to get results, what we're actually looking at, and what that information might be able to provide in terms of additional access to clinical trials and therapies. And so with that, I'll conclude the presentation portion. And thanks a lot for your time today. I've listed here our media contact, Robert Power. That, of course, is not me. But Robert is listed here should you have any additional questions that we either don't get to today or questions that you might think of later. Certainly jot down Robert's email and send him a message. And of course, if you'd like more information about the tests that we offer at Personalis, feel free to visit our website. And we do have a virtual booth here at LabRoots today, so feel free to visit that and ask questions there as well. Thanks so much.
Thank you, Heather, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask Heather Wetzel, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many questions as we have time for. Let's get started. Our first question is, how does the delivery of raw data work? What is provided?
So for raw data, again, I mentioned that we report on those 181 genes and could provide raw data for over 1,300 of those genes on the larger extended panel. And so this is something that we have a consent form in place that would be provided to you that the clinician and the patient would have to sign to be able to access that raw data. What we provide on the DNA side is a FASTQ file, BAM file, and then the sort of variant annotation report. And then for RNA analysis, we're providing the FASTQ file as well. And that can be submitted either via hard drive or FTP. It's sort of whatever works best for in this particular case.
Could this test be used with liquid biopsies?
It's a question that I thought might come by. At this point, we're not accepting liquid biopsies. It is something that we're certainly looking into at Personalis to be able to possibly offer in the future. But at this point right now, the sample types that we're taking are FFPE and fresh frozen tissue.
How can I inquire about certain genes of interest on the reportable panel?
So as I mentioned before, one of the sort of documents that we provide is an actual page, is sort of an information sheet about the CancerPlus test, and on that sheet, the list of genes is actually included there. So if you're looking, I guess if your question is more, is there a gene that's included on that panel, then that would be a good place to look on that particular list that we can provide. If you're more asking, what are the sort of coverage statistics, is this a gene that's well covered on your panel? You could certainly submit that question to us internally, and we can provide some information about sort of the coverage, or maybe even one of those gene coverage plots that I showed earlier on in the presentation to really illustrate the sequencing coverage of that gene.
What is your preferred sample type?
This one's an easy question. So again, we take FFPE and fresh frozen tissue. If at all possible, we'd love a sample from fresh frozen tissue, but also are very happy to take FFPE.
Do you have a sample report that you're willing to share?
We certainly do have a sample report. We don't have one posted on our website quite yet, but if you're interested in sort of browsing through sort of the full report and not just the couple of pages that I showed for the presentation, you know, again, certainly let us know and we're happy to share one with you.
You mentioned optimized coverage of the genes on your panel. How do you do this?
I don't think I have time to go over the whole process here, but just to sort of give a little bit of detail, you know, I've mentioned before that, you know, the process is really sort of this ACE technology, and this is a patented technology, so you could certainly read through all the sort of nitty-gritty details, you know, you wanted to read through the patent itself or maybe some light bedtime reading. But essentially what's happening is really we're optimizing some of the sort of probe design and chemistries that are sort of done prior to the sequencing process to really maximize, you know, our ability to sequence some of those difficult areas like GC content. That's sort of one of the main areas of focus for obtaining that comprehensive coverage.
Is there a minimum tumor content requirement for samples?
Yes, there is. For this particular test, we require 20% tumor content.
Do you call CNVs in all genes?
We don't call CNVs in every single of the 181 genes. We call them in 41 of those 181 genes. And again, that, you know, if you were looking at specific genes and wanted to sort of see what genes we call CNVs in, that would also be provided on that sort of information sheet for CancerPlus.
Where do you get your gene list from?
I think it cut out for just a second, but I think the question was, where do you get your gene list from? And you know, the gene list, again, is a really focused gene list where, you know, what we're looking for is really testing and reporting on genes that are clinically relevant. And so that the vast majority of those genes are genes that have active clinical trials and therapies associated with them. And, you know, there is a smaller subset of genes that are frequently mutated or in cancer, but might not yet have an active therapy associated with it. But the genes themselves are pulled from a number of
I'm not sure if you were finished answering that question, but let me.
Nope, I was finished, Judy.
Thank you, Heather. Sorry about that. You mentioned this was for solid tumors. Do you have plans to offer a similar test for leukemia, for example?
Another question I thought might come up. You know, this is certainly something that we are investigating. We don't have really, I would say, a release date or an actual time frame for when we might offer a test for non-solid tumors, but it certainly is something that we're considering.
This will be our last question of the day here. What is the cost of the test?
So I don't want to give specific pricing information right now, but I will say that the test itself is very competitively priced. And so if you have ordered, you know, a similar sort of tumor profiling test from another laboratory, you know, the pricing would certainly be in line with what those are being offered at. If you'd like to get more specific pricing information and talk about pricing for your institution, again, feel free to contact, sort of our internal media contact, Robert Power, at the address listed below, and we can be happy to give you more information.
I would like to once again thank Heather Wetzel for her presentation. Do you have any final comments?
Thanks, Judy. No, I don't have any further comments. Thank you all for your time, and enjoy the rest of your day. I hope the conference is great.
Thanks again. I would also like to thank our sponsor, Personalis, for making today's webcast possible. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CME CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through October 7th, 2016. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.